Welcome back to this next video, and this is the part 2 on the Salmonella. If I give you a recap of the previous video, I've told you that Salmonella is a genus of rod-shaped gram-negative bacteria, and they are medically important pathogens for both humans and animals. The Salmonella species are non-spore forming facultative anaerobic bacteria, and they are predominantly motile because of the presence of the peritrichous flagella. Now, the genome size of the Salmonella that vary among the serovars with the range from 4460 to 4857 kb, and the Salmonella form a complex group of bacteria consisting of two species. One is known as the Salmonella eকথerica and the other one is known as the Salmonella bungori. It have got 6 subspecies and more than 2579 serovars based on biochemical and genomic modifications. Now, the cycle of contamination, I've told you that the main niche of Salmonella serovars, that is the intestinal tract of the humans and farm animals, but it can also be present in the intestinal tract of the wild birds, reptiles, and occasionally insects. And the route of transmission, that is the fecal-oral transmission. Now, the subspecies eকথerica, that is further divided into the typhoidal Salmonella, and the examples are the Salmonella typhi and the Salmonella paratyphi, which cause typhoid and paratyphoid fever in humans. And the other one is known as the non-typhoidal Salmonella, for short, that is known as the NTS, and it contains S. typhimurium and S. enteritidis, which can actually cause self-limiting gastroenteritis and extraintestinal diseases in humans and animals. Then I've told you that typhoid or enteric fever is caused by the Salmonella typhi and paratyphi A, B, and C, and it is only the Salmonella typhi that or paratyphi that actually only lives in the human. And the route of transmission, that is the fecal-oral route, and the typhoid fever is more established in the developing countries as compared to the developed countries. Then I've told you about the biochemical identification of the Salmonella typhi, that they are gram-negative, they are catalase negative, they are negative for capsule, spore, and gas. They are motile, they are oxidase negative, they produce hydrogen sulfide gas, they are facultative anaerobe. Then I've told you that one of the main characteristics that distinguish the Salmonella typhi from the non-typhoidal Salmonella typhi is the production of a polysaccharide capsule, which is known as the VI antigen, which actually inhibits the phagocytosis by the macrophages, and it confers serum resistance by shielding the O antigen from the antibodies. And the genes for the VI antigen, they are present in on the Salmonella Pathogenicity Island 7. Then the Salmonella have developed this acid tolerance response by the production of the ATP and the FUR genes. Now, in this particular video, I want to focus on a specialized system that is present in the Salmonella typhi that helps the introduction of the virulence vectors from the Salmonella into the host cells. Now, this particular system is known as the type 3 secretion system. It is sometimes written as the type 3 secretion system, and it is abbreviated as the type 3 secretion system, like TTSS, which is the type 3 secretion system, or sometimes it is written as the T3SS, which is the type 3 secretion system. And this type 3 secretion system is also known as the Injectisome. Now, what this Injectisome do is that this Injectisome is going to introduce the virulence factor from the Salmonella typhi into the host cell. So this is the lipid bilayer of the Salmonella typhi, and this is the lipid bilayer or cell of the human host cells. Now, what happens is that when you look at the structure of this type 3 secretion system, the type 3 secretion system is made up of a variety of the proteins. Now, the first component is known as the export apparatus, as you can see over here, that is actually present on the inner membrane side of the Salmonella typhi. And this export apparatus is made up of a variety of proteins, which is known, one of the important protein is the InvA, the other one is the SpaP, the next one is the SpaQ, the SpaR, and the SpaS. So these proteins, these 5 kind of the proteins, they are actually going to make this export apparatus. Now, there are other important proteins, as you can see over here. One is known as the PrgH, and the other one is known as the PrgK, and these protein are actually making the inner ring like structure over here, as you can see here. Then you have got this protein which is known as the InvG, and this InvG is actually present on the outer membrane side of the salmonella, and it is actually known as the outer ring. Now this whole complex, this export apparatus, this inner ring and this outer ring, they are actually working as a foundation for the formation of this rod-like structure over here. You can see over here, and this inner rod-like structure is actually made up of a different protein which is known as the PrgJ. So the names are only written on one side, but the name are true for the other side as well. This one will be the export apparatus, which will be made up of these particular proteins. These two proteins you can see over here. So this is the PrgH, this is the PrgK, this is the InvG. So the outer ring, the inner ring and the export apparatus is actually helping in the formation of this inner rod-like structure. Now on the outer side of the Salmonella typhi, you are actually seeing this needle-like structure, and this needle-like structure is made up of this special protein which is known as the PrgI. Now on the tip of the this needle, you are actually having this special protein which is known as the SIPD, that will be present on both sides, and this SIPD is stably bound at the tip of the needle. Now upon host contact, this protein SIPD, it actually makes a platform for the translogon, and this translogon is composed of the SipB and the SipC. So the SIPD is actually the protein that is in interaction with the host cell, and then it recruits the SipB and the SipC. This SipB and the SipC, as you can see over here, that is actually an extension of this needle, and when this is extended, it is actually present, it is actually introduced into the interior of the cell. Now this whole structure is actually known as the type 3 secretion system, and with the help of this type 3 secretion system, the virulence factor that are present in the salmonella, that are introduced into the human host cell, where they can cause the symptoms of the typhoid. Now the proteins, the different proteins that are making this type 3 secretion system, that are present on a special region of the genome of the Salmonella typhi, and that special region is known as the Salmonella Pathogenicity Island 1. The one we see for the VI antigen, that was the Salmonella Pathogenicity Island 7, but the proteins that are making this type 3 secretion system, that are present on the special region of the genome of the Salmonella typhi, which is known as the Salmonella Pathogenicity Island 1. Now this SP1 encoded type 3 secretion system is required not only for cell invasion, but also for the suppression of early pro-inflammatory cytokine expression in the macrophages. Because what happens is, when the bacteria enters into the human beings, a pro-inflammatory response, a cytokine response is generated by the immune system of the human, which is actually responsible for neutralizing the bacteria. Now what these effector proteins do, or what the virulence factors of the Salmonella typhi do, is that they are actually responsible for suppression of this pro-inflammatory cytokine expression in macrophages. Now, when this cytokine expression, that is when cytokine expression in macrophages that is suppressed, that means that the bacteria is actually having a favorable environment for its establishment in the human host. Now these effectors, these virulence factors or the effector proteins, they are one and the same thing. So these effectors, they play a variety of role during salmonella infection, including taking part in the rearrangement of the host cytoskeleton. And when there is a rearrangement of the host cytoskeleton, that actually favors the entry of the bacteria into the host cell. There is immune cell recruitment, and the bacteria actually use these immune cells for their growth. And there is fluid secretion, which is actually one of the symptoms of the diarrhea that you can actually see in the typhoid individuals, and the regulation of the host inflammatory responses. If I give you examples of the important virulence proteins or the effector proteins that are introduced into the that are produced in the Salmonella and introduced into the human host cell by the type 3 secretion system, one of the important one is known as the AvrA protein. Now this AvrA gene, this is located in the Salmonella Pathogenicity Island 1, and this AvrA protein is actually a multifunctional enzyme, and it plays a critical role in inhibiting activation of the key pro-inflammatory NF-κB transcription factor. Now this NF-κB transcription factor that is very important in generating an inflammatory response, and if the AVAR is actually inhibiting this NF-κB transcription factor from its activity, it means that the pro-inflammatory response that will be reduced or that will be inhibited. This AVAR, it also stop the process of the apoptosis via the JNK pathway. This JNK pathway is actually the Janus kinase pathway. Now, what happens is when this AVAR, it inhibits the NF-κB transcription factor, it is inhibiting the production of the pro-inflammatory cytokines. The bacteria is having a favorable environment for its growth. Secondly, when it inhibits the apoptosis, that means it can easily grow inside the cell and it do not allow the death of the cell. Now, the Salmonella AVR modulates survival of the infected macrophages, likely through the JNK suppression, and prevent macrophage death and rapid bacterial dissemination. Now, this AVRA suppression of apoptosis in infected macrophages may allow for establishment of a stable intracellular niche typical of intracellular pathogens. So, when it is inhibiting the apoptosis of these macrophages, it can actually establish itself inside the infected macrophages. Now, the AVAR expression in Salmonella that also influences the function of the tight junctions of intestinal epithelial cells, and when the function of the tight junctions of the intestinal epithelial cells that is damaged, that actually allows the bacterial invasion and translocation into the host cell. Another important virulence factor that is known as the SIPs. They are known as the SIPs, and there are a variety of the protein within this SIPs family. One of the important one is known as the SipA, and that is an actin binding protein, and it enhances the efficiency of the entry process into the host cells by influencing different stages in the formation of membrane ruffles and rearrangement of the actin cytoskeleton. Now, what happens is that this actin is actually making the cytoskeleton of the human cells, and it is providing a particular integrity and shape to the host cells. Now, what this SipA do is that it is going for the rearrangement of this actin cytoskeleton, and this SipA, it actually regulates the concentration, polymerization and stability of the actin molecule at the site of the bacterial entry where the bacteria have interacted with the host cells, and it increases the bundling activity of the host cell membrane. And by this particular activity, the actin cytoskeleton of the human cells that has been compromised, and when that is compromised, that actually allow the entry of the bacteria into the host cells. Now, the translocation of the SPTP to host cell results in the disruption of the cellular actin cytoskeleton, and this SPTP is another important member of this SIP family, which actually results in the disruption of the already established cellular actin cytoskeleton. One of the important toxins of the typhoid that is known as the typhoid toxin. And this typhoid toxin is actually this typhoid toxin is actually responsible for the death of the intestinal cells. And when there is death of the intestinal cells, that means there will be no absorption of the food and there will be diarrhea. If you talk about the structure of this typhoid toxin, it is actually a pyramid-shaped complex. As you can see over here, you are actually seeing a pyramid-like structure, and this pyramid is made up of PLTB, 5 molecules. There are 5 molecules of the PLTB. There is 1 molecule of the PLTA, and there is 1 molecule of the CDTB over here. Now, this PLT is actually the pertussis-like toxin. So this will be the pertussis-like toxin A, this will be the pertussis-like toxin B. So is these toxins have got similarity with the toxin of the pertussis, therefore they are known as the pertussis-like toxin A and the pertussis-like toxin B. And then you have got this endonuclease part over here, which is known as the CDTB over here. Now this CDTB, it is actually the cytolethal distending toxin. It is one of the important component of the typhi toxin. Now what this CDTB do, this CDTB, what it do is that it is believed that it initiates DNA degradation, which causes cell to arrest in the G2 stage of the cell cycle and subsequently die by apoptosis. Now what happens is during the cell cycle, when there is damage to the DNA, what the p53 protein do is that they stop the cell cycle. First they attempt to repair the DNA by the DNA repair system, but if the DNA damage is so severe that it is not repairable, then the p53 signal the process of the apoptosis to remove that cell from the body. Now what happens during the typhi toxin is that when the CDTB, when it starts the degradation of the DNA, the degradation is so intensive that the DNA repair system is not able to repair it, so the process of the apoptosis starts, and hence the cell that has been infected by the typhi toxin that die out. Now it has been hypothesized that this particularly occur in the crypt structures of the intestines, where cells are normally rapidly dividing. Now the death of these specialized cells is thought to lead to the loss of function and erosion of the protective intestinal epithelial layer, thus producing the electrolyte disturbance and malabsorption, which is associated with diarrhea in the typhoid disease. Now what happens is how this typhoid toxins it enters into the epithelial cells, or how it interacts with the epithelial cells. Now in epithelial cells there are the typhoid toxin. There are special receptors for these typhoid toxins, which is known as the podocalexin-like protein 1, that is also expressed in vascular endothelial cells. Now in contrast, in macrophages and the T and B cells, the receptor is CD45, which is ubiquitously expressed in all hematopoietic cells and other erythrocytes and platelets. Other than erythrocytes and platelets, the CD45 receptor, it is not present on the erythrocytes and the platelets, and it is present on all of the other hematopoietic cells. Now the typhoid toxins that actually recognize the common glycan moieties on these heavily glycosylated surface proteins. So when you talk about these epithelial cells, the receptor, this PODXL, it is actually having glycan moieties over here, it is having carbohydrate moieties over here, and the typhoid toxin is actually recognizing those glycan moieties on the in this particular receptor. Similarly in the CD45, there are glycan moieties, and those glycan moieties that are recognized by this typhoid toxin, and when they recognize this typhoid toxin, they are going to interact with each other, causing the condition of the diarrhea. If you talk about the signs and symptoms of the typhoid, so the incubation period for the Salmonella typhi is usually 5 to 14 days. People usually have a sustained fever. By that I mean one that doesn't come and go. There is sustained fever, and that can be as high as 103 to 104 Fahrenheit. There is weakness, there is stomach pain, headache, diarrhea or constipation, the cough, there is loss of appetite. Now some people with typhoid fever or the paratyphoid fever, they develop a rash of flat rose colored spots on their body. There are red spots on their body. In some people you will see this rash of flat rose colored spots on their body during the typhoid fever or the paratyphoid fever. If you talk about the diagnosis, so the gold standard for the diagnosis of the Salmonella typhi is to go for the blood or stool culture. And the most common media that are used in this stool or blood culture, one is known as the Salmonella Shigella agar. As you can see over here, the Salmonella is going to give you these black colonies that you can easily see, and this Salmonella Shigella is actually a selective as well as a differential media for the growth of the Salmonella. And then you have got this another important media which is known as the Xylose Lysine Deoxycholate. So you can use both of them because both of them, they are commercially available and they are acting as the selective and differential media for the growth of the Salmonella typhi. So the gold standard is the culture for the blood or the stool culture. Another important test that is known as the Vidal test, and this Vidal test is actually going to recognize the antigens which are present on the surface of the bacteria. As you can see over here, you can have this H antigen which is actually present in the flagella of the Salmonella typhi. You can have this O antigen which is present on the surface of this particular bacteria, and then you can have this VI antigen which is also present on the surface of the bacteria. So the Vidal test is actually going to recognize these antigens. Now what happens is that you have to take the patient, you have to take blood from the patient, you have to isolate the serum from that particular blood, and then you have to go for the serial dilution of the patient serum. Like this is the phenomena which you actually do for the serial dilution. So if this is the, say for example, the initial sample that you have taken, you have to introduce one drop over here, and then you have to take one drop from here and you have to introduce that into the other, and so on. So you are actually going to dilute the sample of the patient serum by this phenomena of the serial dilution. After that, you have to eat equal volume of the Salmonella antigen, and the common antigen that are detected, they are the O antigen and they are the H antigen. As you can see over here, this is the serum of the patient in the dilution of 1 to 20, 1 to 40, 1 to 80, 1 to 60, and 1 to 320. So you have to add the Salmonella antigen to all of these dilutions, and then you have to go to check for the agglutination, because the Salmonella antigen, if the Salmonella is present, that means that the antigen that will be present in the serum of the patient, there will be antibodies, and when the antibodies they interact with these O antigens, that means there will be the agglutination phenomena. So you have to check the agglutination phenomena over here. For example, if you are getting agglutination in the 1 into 160, that means this is usually considered as the titer, and this titer in this particular case, that will be 1 to 160. So the dilution at which the agglutination start, that is actually considered as the titer. So if you talk about the interpretation of the Vidal test, so when you go for the reading of the test, the highest dilution of the serum is noted where there is agglutination. If it ends in 1 to 160, as you can see over here, that means that will be considered as the titer. Now, the Vidal test is positive if the O antigen titer is greater than 1 to 160, and if it actually show you the active infection. So if the antigen titer for the O antigen is greater than 1 to 160, that means it is an active infection. If the H antigen titer is greater than 1 to 160, it indicates past infection or in immunized persons. If you talk about the O antigens, now this O antigen in the acute stage, the antigen O will be positive and the titer will be more than 1 to 160. Now O antigen appears early and it also disappears early. If you talk about the H antigens, the H antigen arises late and it disappears late. This will be positive in the recovery stage. When you talk about this VI antigen, this is actually as indicator of the carrier stage. If we talk about the treatment of the Salmonella typhi, so the treatment is usually the use of the antibiotics, and the drug of the choice is chlorphenicol, but it has got many side effects. So now the drug of choice is actually the ciprofloxacin. Now the ampicillin, amoxicillin, third generation cephalosporins, and the fluoroquinolones that can also be used for the treatment of the Salmonella typhi. And clotrimazole is better than the chlorphenicol because it have got less side effects. There are a variety of the antibiotics that can be used for the treatment of the typhoid fever. If you like the video, please subscribe to my channel, hit the like button and share it with your friends.